Selective breeding. We often like to talk about selective breeding when we discuss genetic engineering because it is an old style of choosing traits with organisms in order to get a desired outcome. It's just a little bit more of a low-tech way. So during selective breeding, we're choosing organisms with desired traits in order to breed them together. The idea is our whole goal is to increase genetic variation or in hopes of getting certain traits. Farmers, breeders have been doing this for hundreds and even thousands of years when we go back to early farming days. Farmers have selected plants and animals with desired traits, crossed them together in order for a hope of having a better crop or a better animal in the outcome. So there are two main things that we talk about. We talk about selective breeding. The first one is called inbreeding. Inbreeding is basically when you take two organisms that are very closely related to each other. Maybe they have similar parents. If you're talking about breeding animals together, they may be third in line with respect to maybe something like cousins. In this case, you will enhance similar traits that that line particularly has. It ends up decreasing genetic variation. The idea is to get more traits that are similar to each other. This happens a lot in purebred dogs. In purebred dogs, sometimes you don't have a huge genetic pool to choose from, so if you start breeding animals that are relatively closely related to each other, they will have similar traits. You'll decrease the genetic variation, something we call inbreeding. Another form of selective breeding would be called hybridization. During hybridization, you're combining different traits. You're taking two animals that maybe aren't necessarily very close to each other or don't necessarily have similar traits in order to increase genetic variation. We have this happen in things like dogs. When we combine a Labrador and a Poodle to create a Labradoodle, we tend to get this mix. We don't necessarily want to call them mutts because it was an intended breeding. And the idea is that they usually want the personality of a Labrador, which is pretty happy and easygoing, and a Poodle, which sometimes doesn't necessarily shed quite as much. The goal is to have those traits combined. It doesn't always work out quite so well, though. The liger, a cross between a lion and a tiger, it ends up with a larger animal than the lion and the tiger individually, and we use that in tomatoes as well to end up with a better tasting tomato, a tomato that maybe can resist pests, things like that. When we do hybridization, we often get something called hybrid vigor. Sometimes it turns out that the mix from the two original individuals happens to be, we would want to say, more fit or stronger in the environment than the original parents were. There are some cons to selective breeding. It does take a long time. Sometimes it may take 10, 20, 30 generations of breeding in order to get your desired outcomes. In some cases, especially with the case of inbreeding, you get a lot of unwanted traits. Since you're breeding animals that are relatively closely related to each other, you can get an increase in genetic mutations that tend to maybe run in that line, or genetic disorders. And the other problem is you don't always get your desired traits. For example, when you're crossing that Labradoodle, your goal is to get the personality of the Labrador and the pluses of maybe a Poodle that doesn't shed quite so much, but we don't always get those desired traits in the outcome because we know that all of these organisms are reproducing sexually. The chances of getting those traits in the offspring are really not necessarily a given every single time. Another thing that we can do with genetic engineering is create what's called a transgenic organism. This is where we are actually taking an organism with genes from two or more species. So we're creating an organism that has genes from species that normally cannot breed. So this is something that can only happen in the lab. Artificially, this wouldn't happen in nature. We know that two different species can't interbreed and create viable offspring with each other. So we have some examples of what you could do with a transgenic organism. What are some examples of what we would want to use it for? We could create an animal with extra growth hormone. So maybe that animal would grow faster and leaner. They've done this with salmon. They've crossed salmon with different types of fish, inserted the DNA from those fish for the gene that allows them to grow faster and grow for a longer period of time in order to create salmon that are much larger than a traditional salmon. Plants with pest resistance. We have created some plants that naturally create their own insecticide, let's say, their own chemicals, that would allow them to not be damaged by pests. In this case, it's beneficial for the farmer growing the crop because they don't have to spray pesticides. It's also a plus for the environment because we have fewer pesticides being sprayed. Another example would be bacteria that create or have a human gene for growth hormone in them. The idea is then that the bacteria would make the growth hormone. The nice thing is we can do this with other genes as well and have the bacteria actually create whatever it is that gene is supposed to produce. In all cases of transgenic organisms, we would call these genetically modified organisms. A lot of times they're abbreviated as GMOs. A couple of things to think about with GMOs. Do you agree or disagree with them? Do you agree with the idea of taking two organisms that maybe wouldn't normally be able to interbreed with each other and artificially selecting traits to combine together to make this new organism that has traits that never would have been possible in that organism all on its own? In some cases in the past, they've taken tomatoes and they've taken a gene that comes from fish that allows it to survive very cold waters and put that gene into the tomatoes to survive frost and very cold weather. Think about some of the pros and cons that come along with creating transgenic organisms. We're going to discuss these issues in class and sort of come up with a little bit of a debate for ourselves. Let's see how we actually specifically go along and create a transgenic bacteria. So let's say we have a bacterial cell. Remember that a bacterial cell is prokaryotic. It doesn't have DNA contained in a nucleus, so sometimes that DNA is just floating around in the cytoplasm. There are some bacteria that also have a circular-shaped DNA or a ring-shaped DNA, and we call that a plasmid. So scientists have figured out how to remove this plasmid from the bacterial cells, and they can cut open that ring of DNA or genetic material with special enzymes called restriction enzymes. They recognize specific sections of DNA and cut it at that section. What it essentially does is creates an opening or a gap in that DNA. What we can do then is we can take a human cell, which remember is a eukaryotic cell, has its nucleus with the DNA contained inside a membrane-bound nucleus. We can remove the DNA from the nucleus. We can use similar restriction enzymes in order to isolate, identify, and remove a particular gene. Let's say in this case, the gene that produces insulin to control your blood sugar. So let's say we've removed that gene. We can now use another set of enzymes, almost like a glue, to glue the human gene for insulin production into that open segment of the bacterial plasmid DNA. We can now join them together, so it's a ring again. Now we have what's called a recombinant DNA structure. We've recombined DNA from two organisms or two species. We then can insert the DNA back into a bacterial cell. And the neat thing is we know that bacteria reproduce by mitosis. So those bacteria, every organism that they reproduce is going to have that gene in it. And each of those bacterial cells now have the gene for insulin production in it, and we know that they're going to be producing insulin. The neat thing is they're producing human insulin, not insulin from another organism. We can then use that for people who have diabetes, and instead of getting insulin from other organisms or having to harvest it or create it in a laboratory. The last example we have for genetic engineering is cloning. Cloning is when we create an exact genetic copy of an organism. So there's one way to do cloning that's relatively simplistic. What we do is we take a donor body cell. Remember, we want to take our body cells, not our sex cells like a sperm or an egg, because they have the entire set of DNA in them. And we want to copy that exact person, so we need all their DNA. So we're going to take a body cell from a donor, the person that we want to clone the DNA from, and we're going to extract the nucleus. We're going to get the DNA out of the nucleus, and we're going to combine that with an egg cell from a donor. Now, the egg cell donor, all we want is the egg cell from them. We don't want their DNA. We're going to get the DNA from our other donor. So we're going to remove the nucleus from the egg cell, so we just end up with a shell of an egg, not the nucleus inside. We're going to fuse the DNA from my donor and the egg. to create what we would consider almost like a fertilized egg, except it didn't get fertilized by a sperm. We're going to use some special hormones and electrical shock to force that cell to think it's undergoing cell division and mitosis, so it'll start dividing just like a regular embryo would and a regular cell, and eventually we can implant that embryo into the uterus of any other organism of the same species, which we would call a surrogate. They would basically just be holding and growing that embryo, let it go through a normal gestation length, and eventually we would have a newborn, and that newborn would be the clone of our original donor of the DNA. They would have an exact copy of the DNA from the donor. So if you think about some natural ways that we've had cloning, asexual reproduction in organisms that are a little bit more simplistic, like bacteria, amoeba, paramecium, and we've also had identical twins. Identical twins happen naturally, and they have exact copies of DNA from each other. It's a natural way of cloning. It's just not a way that we can control or predict.